will start the action potential. Okay? Like I said, a lot of it, believe it or not, is going to come from higher order thinking skills, which is really cool. But we'll get into that later. Now, once we can get the action potential, as the action potential travels down the axon to this terminal end, note that we're going to have some vesicles of acetylcholine available. Now watch, watch what happens to the acetylcholine. The acetylcholine, it's wrapped in a little vesicle. The vesicle fuses to the membrane right there on the nerve, releases its substance. Now, to get the acetylcholine to be released, do you notice anything?
Calcium.
Calcium. I had to have calcium to get the acetylcholine to be released. So that tells me right now for a muscle contraction, calcium is needed. All right? Now once I have the acetylcholine get released, the acetylcholine diffuses across this synaptic cleft, and notice how they're representing a ligand-gated, all right? And the ligand-gated acetylcholine matches the receptor site. The receptor site accepts the acetylcholine. Sodium that's outside the plasma membrane gets to rush in. Now, if I had thousands of those types of receptors located in this region, think of all the sodium that gets to go across that plasma membrane. So my sodium gets to flow across the plasma membrane. Now, the sodium flows in. From this point down, the muscle fiber, okay, what gates are going to be present? Voltage gated. And then I need what to get it back into balance? My sodium potassium. Now, the acetylcholine that got released, the acetylcholine, we can't have it just simply stay in that cleft because it would continue to be accepted by a receptor, and the receptor would continue to let sodium in. So what we're going to find is we're going to have acetylcholinesterase, the enzyme that will break acetylcholine down. Now watch what happens to the acetylcholine. The acetylcholine can be broken down into the choline. And what do you see the choline do? It crosses back over the cleft and moves back into the terminal end of the nerve. And then it will go back up the nerve, and the nerve can reuse it to make more acetylcholine.
So what happens to the calcium?
The calcium gate will close, and calcium will just go back out.
It just goes back out?
Yeah.
It can come back in then?
Now, yes, the calcium will flow back across.
Okay.
Took me a minute to think about that. Yes. The acetyl portion of acetylcholine, have you ever heard of acetic acid? What is it? You've heard of it, I know you have. It's vinegar. So it simply becomes acetic acid, and the body will degrade it and get rid of it. Is that not the coolest process ever for muscle contraction? So we've got to have all of this working properly, and the muscle, if the signal is strong enough, will create the contraction. Now note, this is occurring on one fiber. And if we were looking at this muscle, for example, okay, remember, fiber, 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 fiber. Because fibers are going to be into fascicles, the fascicles are going to make up the muscle. Remember all that? Okay? Each one of them having their nerve, each little fiber. So if I've got a nerve coming into this, and that potential coming down goes to all these fibers right here, what are the chances of making this muscle contract? Good or bad?
Good.
Good. Does that make sense? So, when the muscle will contract, we call this excitation, contraction, coupling. Coupling. What does it make you think about? Huh? What? Pairing something? I think about plumbing. A coupling that's needed for pipes. Okay? Go figure, right? Excitation, contraction. We had excitation from the nerve, contraction from the muscle. The two together, excitation, contraction, coupling. Does that make sense? So this is what you'll see it referred to as. You'll see it referred to this action, okay, potential that we get from the nerve and the contraction of the muscle. Look at everything that's involved. The sarcolemma. What is that? Men? It's the sarcolemma is the term for the plasma membrane on the muscle cell. Okay? So sarcolemma is the term for the plasma membrane on the muscle cell. We're going to have these things called T-tubules, transverse, running through the muscle fiber. They're little invaginations of this sarcolemma, this membrane. And note how it looks like they just make a cut straight through. Y'all share. Right here. And since y'all are up here with me, we'll share this one. Okay? See this blue stuff? Okay? See the blue stuff? Notice how they're trying to represent it here, 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 here. But here, they're showing you how it will cut straight through because it's transverse. Okay? That's your T-tubule. All right, is everybody with me on the T-tubule? Now look at what they're trying to represent in between them. See this kind of webby looking stuff? Okay? We're going to have terminal cisternae, we're going to have sarcoplasmic reticulum. Now, when we get a T-tubule and terminal cisternae located beside it, we're going to refer to it as a triad. Now, the structure that we're being explained, look at it on that. My T-tubule, making that cut all the way across or through it, okay? Terminal cisternae, all right? Got all this webbing that goes around it. Sarcolemma is that plasma membrane. This kind of like pouchy looking stuff in between is going to be sarcoplasmic reticulum. Now, we've normally thought about endoplasmic reticulum, right? Is that sort of what it makes you think about? Because it's this, well, it's like the cistern, okay? It's like a cistern-y stuff, all right? That little cistern, and then all this stuff in between, the reticulum, okay? This transverse tubule, this is an area for storage. This little webby looking stuff, okay? This is an area for storage. Can you guess what it's going to store? Huh? Calcium. So now I've told you that we've got the calcium in that nerve to make the acetylcholine get released. And I've now told you calcium is going to be stored in that webby looking stuff, the reticulum. Okay, is everybody with me? Troponin. Huh? What? What was the troponin? The little 3 subunit thing stuck on the tropomyosin, right? All right. Thing probably wasn't the correct term to use. Units. Okay.
3 subunits stuck where?
Remember how we had the chain around the actin, and at every bend there was those 3 little pieces? That was troponin. The 3 little units. So, this coupling that we're going to be able to have between a nerve and the muscle that it innervates, we have to get to the level of that sarcomere and my troponin, my actin and my myosin. Okay? Yes, no, maybe. Follow along with me, okay? Nerve meeting the muscle. Action potential comes down. Acetylcholine gets released. We open those ligand gated. Sodium rushes in. From that point, we're dependent on the voltage gated to travel the rest of the fiber, correct? All right. Now, that fiber, the functional units are what? the sarcomere. These transverse tubules